Hey guys, welcome to my channel. Just want to let you know that we're here today to talk about a passive Pisces. And also we're going to be going over a few basics for phlebotomy before I begin my phlebotomy tips and tricks videos. My passion for healthcare is unlimited. If I had the brain power as strong as my drive, I would become certified in all things medical. Seriously, my dream began wanting to be a firefighter paramedic back in high school. But in 2017, I ran across the phlebotomy course, and a fire just was kind of lit within me. I felt this spark before in my past, but this was different. So I made this decision once I started and realized quickly that I was actually very skilled in this area, and I wanted to teach others and build their confidence within themselves. My goals are to go do it via vlogging. I will be teaching tips and courses over time until I can fully open my own online school, which I feel is going to be completely cool. What is phlebotomy? It is derived from the Greek words phlebo and tomi. Phlebo relating to veins, and tomi relating to cutting. Phlebotomy, incision of the veins and blood collection, or basically cutting of the veins and blood collection. One thing you should always remember and do not forget is the phleb, your order of draw. Your glass vials, which are your blood cultures, and they are completely sterile, should always go first. Then you have your yellow red tubes, sometimes glass, sometimes plastic. Those are your SPSs. They should go next. Before anything else, you should always do your light blue tubes. Those are your citrate tubes, and they should be filled completely. Never ever underfill them. Then you have your speckled red. Here we have your three main veins. Now you must know that your first vein you should always try to hit is your median cubital. That is the middle vein in the arm. Now it should always feel bouncy and ready to be stuck. If you don't feel that bounce, it's most likely not your best choice to take. So then you would go on to your second, which is your cephalic vein. That is the outer vein, which is towards the outer portion of the body. So remember, when you're saying the outer vein, it's towards the outer portion of the body. Next is your basalic vein. That is your inner vein, which is towards the inner portion of your body. So that's the last one you want to try. After that, I would not stick any more unless necessary, or go into the hand with the butterfly. Other than that, you should not do any other veins, because you do not know if you would hit an artery or if you can cause collapsed veins or infiltrations. Your blood types. Everybody kind of wants to know a little bit something about that. You should always know your blood type. They are determined by the absence or presence of antigens. These are found on the surface of the red blood cells. The Rh factor is also a very important factor that is either absent, negative, or present, positive, making 8 in total for your blood types. AB has both A and B antigens on red cells, but neither the A nor the B antibody is in the plasma. The A has only A antigens on red cells and B antibodies in the plasma. B has only B antigens on red cells and A antibodies in the plasma. O, it has neither A or B antigens on red cells, but it has both A and B antibodies on the plasma. So basically, you have all 4 of these blood types, and if you were to have the absence or the presence of the Rh factors, they would become AB positive, AB negative, A positive or A negative, B positive or B negative, and then you have O. As I spoke in our order of draw, after those 6, Then and only then can you draw other tubes. So you have your sterile tubes, which you do not invert 0 times. Do not invert them. Then you have your light blue tubes, your citrate tubes. Please invert those only 3 to 4 times. That is the only one that is inverted 3 to 4 times. Your light blue citrate tube. Then you have your tiger tops and your gold or your plastic red tops. Those are all inverted 5 times. Please make sure that you only invert them up to 5 to 6 times. Your green, there is dark green and there is light green. Make sure that they are inverted 8 to 10 times. Same for your lavender, and then your gray pearl tubes are inverted 8 to 10 times also. So a good way to remember it is the first tubes you would pull, your sterile tubes, 0. Then you have 3 to 4. Then you have 5. Then you have 8 to 10 for the last 3. The blood specimens that are used for analysis. These are basically the tubes that we take, and this is the specimens that come from them. You first have your whole blood samples, which anticoagulant is used. They are used for blood typing, cell counts, and determining hormones and metal levels. Then you also can get your serum from those tubes. There are no anticoagulant used. Fluid-filled portion of the blood that remains after the clotting happens. It takes 30 to 60 minutes for the clot activation time. It is mostly used in blood chemistry and immunology. Third, you have your plasma, when you put it through the centrifuge. This is the fluid portion of the blood that includes fibrogen and other clotting factors. Remove the whole blood using anticoagulant and a centrifuge. You collect it for coagulant studies, also used for stat chemistry studies when it is no time to wait for the clotting to occur. These are important tests that you should know, not tests, but the important analysis that you should know. Here you have your common blood tests. Now these are the tests that are usually commonly drawn through those speeds that you take. You have your complete blood count, your CBC, your white blood count, your WBC, your differential, your diff, your platelet count, your mean platelet volume, your red blood count, RBC, your hematocrit, your HCT, your hemoglobin, your HBG, your mean corpuscular HBG, your mean corpuscular HBG concentration, your mean corpuscular volume, and your red cell distribution width. These are common tests that are usually required in stat labs or non-stat labs. Hematology is the study of the function and natures of diseases of blood and of organs that form blood. Hematology is the treatment of blood disorders and diseases, including types of hemophilia, leukemia, lymphoma, and sickle cell anemia, and etc. Another very, very key important part of understanding and being a phlebotomist is your circulatory system. The circulatory system is made up of blood vessels that carry blood away from and towards the heart. Arteries carry blood away from the heart. Arteries away. Pathophysiology, one of my favorites. Pathophysiology is the study of the changes of normal functions within the body and the mind, either caused by a disease or resulting from an abnormal syndrome. Basically, in layman's terms, it is basically the study of the changes within your body and mind when a disease or something abnormal has happened to you. So it's basically studying those changes. You get to learn from it from the cellular function all the way up. It is very interesting to me. Sorry, neat nerd moment. Okay, so basically everything in this slide has been very, very important. But this is one thing you should always want to know. Your basic vitals. You want to always check someone out before you do take their blood. So vitals are always, always very essential. Your BP for your systolic should always be between 90 and 180. Your systolic should always be between 50 and 100. And your pulse should be between 50 and 100 also. The hematocrit for a female should be between 38 and 54, while the hematocrit for a male should be between 39 and 54. The total protein should be between 6 and 9. The temperature. So guys, this has been my first video that I have done on phlebotomy. I have started with the basics of phlebotomy. I will be coming back with your phlebotomy terms and some more on some phleb basics. So please go ahead, ring the bell, subscribe, like, and share, guys. A Passive Pisces. We come in a lot of different areas, from mental health to foodies to family, all the way back to phlebotomy fun. So please subscribe and ring that bell, guys.